The overall goal of this procedure is to express surface proteins on Mycobacterium using surface decoration with recombinant proteins as an alternative to traditional genetic-based approaches.
These methods can help address important research questions in the field of mycobacteriology, especially in the area of host pathogen interactions and the vaccine development. This technique can be used to screen for potential virulence factors or selected antigen candidates. It can be developed relatively rapidly compared to fastidious and time-consuming traditional genetic-based approaches. Demonstrating this procedure will be Amina Talal, research assistant in my lab.
This portion of the protocol begins with transforming P17 Avidin OVA plasmid into E. coli BL21. To do so, induce 250 milliliters of E. coli BL21 culture with IPTG for 3 hours at 37 degrees Celsius. After 3 hours, lyse the bacteria to solubilize the inclusion bodies by first pelleting the induced BL21 culture and discarding the supernatant. Then, resuspend the pellet in 10 milliliters of lysis buffer and allow the cells to be lysed for 15 minutes. Then, continue the incubation at room temperature using a rotator for agitation and let it go overnight. In the morning, centrifuge